for tonight is going to be Susanne Lee-Baldassini. She used to work on the inpatient floor, so I got to know her there. She's just outstanding, and we're very fortunate to have her come to work with the melanoma program full time. So with that, I will turn this over. I think you.
Do I need to put this on? Or?
I think you're better off with that than with that.
Okay. Just trying to put this on. Let's see. How does this go? There we go. Okay. Well, it's nice to see so many people out this evening. Again, I'm Susanne Lee-Baldassini, one of the oncology social workers here at Smilo, and it's a pleasure to be here. I see some familiar faces from our support group, and tonight I am going to be talking about our resources here at Smilo. Okay. So Smilo is fortunate to have a dedicated team.
You can also use this if you want.
All right, thank you, Dr. Harriet. A dedicated team of master's level oncology social workers, and we're available to you and your family throughout the cancer journey. Our commitment is to provide comprehensive and compassionate care. Our role is to help you and your family cope with the illness, discuss issues and concerns, and connect you with a variety of hospital and community resources. There are a range of resources to assist both patient and family. Now, a word about our oncology support group. Support groups, we know, provide a setting in which cancer patients and families can talk about living with cancer with others in a similar situation. Social support can help you develop a range of coping skills. There are different support systems out there, as we know, including community support groups, online cancer support groups, networks, forums, and charitable organizations. Some history to our support groups. In the 1970s, we saw an emergence of encounter and peer support groups, and more studies on group intervention began to appear in the literature. Then in 1989, a landmark study was published by Dr. David Spiegel, MD, et al. The study looked at 10-year follow-up in women living with metastatic breast cancer. And those are women who participated in the support group for at least a year. They found that support group participants survived almost twice as long as those in a control group. Dr. Spiegel's work offered the first evidence that participation in support groups may intervene in the disease process, and the results received extensive press coverage. So that's a while ago. But since then, demand for support groups from people living with cancer has grown. We have our own support group here at Smilo. It's offered on the third Tuesday every other month, 5:30 to 7 p.m. We just had one, our September group, but the group is offered January, March, May, September, and November. And our group does receive support from the Melanoma Foundation of New England, whose goal is to provide support to melanoma patients, education on sun-safe behaviors, and funds for research. So, why join a support group? Sometimes that's kind of an obvious question, but it is good to just revisit that support groups do usually bring together people with common interests and experiences. People who have been through or are going through a similar set of experiences can sympathize with you, but they can also relate to what you're experiencing and help you feel like you're not alone. That's really kind of the takeaway, one of the takeaways with a support group. Those who do participate in a support group believe they can live happier and healthier and with less distress when they spend time relating to others. And again, evidence suggests that support groups can improve the quality of life with people living with cancer. We do, just as a sidebar, we do have other support groups as well. Sometimes people are coping with more than one cancer, so if you are interested in other support groups, we have a number of disease-specific support groups. As far as the individual, couple, and family counseling, again, licensed social workers are also available to meet with you or your family individually to assess your needs, provide short-term counseling, including crisis intervention. And there can be different points, again, in the journey, sometimes at the point of diagnosis, sometimes if there's new information, or if there are decisions further along the journey that have, discussions that have to be happen. We are available for those discussions and to help patient and family. If there are ongoing needs, we'll work with you to identify local mental health professionals in the community or programs to address the need. So the Yale New Haven Hospital Social Work Department, I've just listed the number if you wanted to jot that down, or if you're interested, we can certainly get that information to you. Additional psychosocial needs. So social workers are also available as a resource when there are other questions that arise related to insurance coverage, copay assistance eligibility, community programs such as Look Good, Feel Better. That's a program that's co-sponsored by the American Cancer Society and Smilo. Transportation resources or other needs such as support for caregivers, talking with other family members and colleagues, negotiating time off from work, applying for entitlement programs such as Social Security, Disability, SNAP, or other programs. We have another program here at Smilo, a very successful program that we've been offering to patients and families now for over 5 years. It's called PACT, Parenting at a Challenging Time. It offers guidance and support to parents with cancer who are concerned about the impact of their illness on their young children. So we provide insight to the parents using really a parent guidance model versus a child guidance model from a developmental perspective on what to expect from children, how to talk with them, as well as clarify how they are coping with the illness. Smilo also is very fortunate to have the Patient and Family Resource Center, and this is a resource center on the first floor of Smilo. It offers a comfortable and quiet environment where Smilo Cancer Hospital patients, families, and caregivers can come to find information, browse the internet, attend workshops, and ask about new and ongoing community resources such as the complementary services, lodging, and more. Our survivorship program. New treatment options and innovative therapies are helping cancer patients to live healthy lives long after their cancer has been treated. The growing number of cancer survivors requires new resources and healthcare specialists to care for the needs and concerns that cancer survivors may have, including medical, social, and emotional worries. There's a clinic that meets here at Smilo weekly, Wednesday afternoons on the first floor, and also in the care centers. We have a number of care centers in the greater New Haven area, and also in Torrington. Complementary services, we have a wonderful complementary services program. This provides patients and families with on-site access to Reiki, massage, yoga, meditation, and other community events designed to improve mind, body, and spirit. There's a phone number to call, 200-6129, and they can answer questions or set up an appointment if you are here in the clinic or if you are inpatient or just want to learn more about what's happening. Palliative care. This is an interdisciplinary team dedicated to providing an extra layer of support to patient and family. The team's focus is mostly on reducing the severity of disease symptoms. The goal of palliative care is to prevent and relieve suffering and to improve quality of life of those facing serious or advanced illnesses. So best thing to do is to talk with your medical team to decide if this is the right option for you. This is a list of, including AIM at Melanoma, of resources, and if anyone is interested in getting a copy of this, I'm sure we can find a way to share that. So just to summarize, as you can hear and see, we do have many cancer support organization programs and services. As social workers, we're here to guide you to those resources that will help you stay informed, give you support, and alleviate distress. If you have any questions for me, you can contact me directly. That's my phone number, my voicemail, 200-2120, area code 203. Or if you're in another part of the hospital or clinic, you can also just ask your nurse to speak with either the clinic social worker or the unit social worker, and we'd be happy to answer any questions. Okay. Thank you.
questions for Suzanne?
Okay,
Thank you.
Dr. Sznol.
I just want to emphasize that, did you take the...
Oh, I do. I have it.
Yeah, just go ahead and give it to... I just want to emphasize how important Suzanne's role and the role of social work here is in the hospital. I mean, many people are fortunate to have significant others, they have the resources to receive care, but there are people that we see in the clinic that aren't as fortunate, that don't have those resources, don't have significant others, need supportive services, because we can't deliver medical care unless people have the infrastructure in order to get back and forth to the clinic, or if they need rehabilitation or something other than Suzanne. And the hospital in general provides those services for us so that we can actually really take care not just of the cancer but of the entire patient. So it's really an important thing, and we couldn't survive without the support of Suzanne and her colleagues. So the next person that I want to introduce is Dwain Fehon. Dwain, I think, was the first person that Steve invited, right, to join our group as a psychiatrist to help with the social, the adjustment needs of patients with cancer. And since I think you've ultimately sort of left our group and gone on to bigger and better things in the palliative care group. But Dwain is going to talk a little bit about coping with melanoma and melanoma resilience. Dwain.
Thank you very much. It's a pleasure to be here. Let me just set up the computer. So I'll be talking about resilience. But before I start my talk, I'd like to ask you all a few general questions. Does anybody know that today is the first day of autumn? Show of hands. Oh, lots of you. That's good. It is. Today's the first day of autumn, the vernal equinox, right? 12 hours of light, 12 hours of darkness. What are your thoughts about autumn? If I were to ask you to think about autumn, what does that make you think about? Winter's coming.
Or goodbye to the heat of the summer.
Goodbye to the heat of the summer, right.
Yeah, getting rid of the humidity in the summer.
Yep, less humidity, those nice fall, crisp days. Pardon me? Oh, right. Yeah, less light. Yes. Right, beauty, color. Right. Yes.
Really good apples.
Really good apples, exactly. Right. What are the emotions that you have about autumn being here? How does it make you feel? I imagine some of you are excited about the fall colors, the delicious apples, the less humidity. Some of you may be thinking, oh, good, I feel better, no more heat. Maybe you're grieving the loss of summer. You wish it were still here. Maybe some of you are looking forward to the winter. Maybe some of you are dreading the winter and the cold. I ask you these questions because, how we think about events in our life, even the change of seasons, has a huge impact on how we think about these things, right? Now, Ben Franklin was quoted as saying that the only certainties in life are death and taxes. Well, I would say that there are probably at least a couple of other certainties, and one of them is that change. Everything changes, right? Nothing stays the same in life. We change, the seasons change, we grow, we learn, we have babies, our families grow. There are great changes that happen in our lives. There are changes that can feel very challenging. The other truth is that life can be stressful, and stress is a part of life. It's inevitable. And again, stress can be associated with many positive events. I'll never forget the wonderful stress that I experienced when my daughters were born and how much change that resulted in my life. But there are also many other things, obviously, that are stressful too, that are harder to cope with. So there are positives and negatives, but everything changes, and stress is a part of life. And certainly cancer is one of those stressors that you all know about, where the experience of having cancer can feel like a never-ending roller coaster that can change how you think about yourself, your life, your future, can challenge your ability to adapt and challenge your ability to cope. But you know, it's only natural, right? It's a very stressful thing. And in fact, nearly everyone who is diagnosed or living with a diagnosis of cancer experiences some emotional distress at some point. This is true for family members as well. Indeed, 35 to 45% of newly diagnosed patients or patients who have a recurrent illness show at some point significant levels of emotional distress in terms of anxiety or symptoms of depression. But you know what? The interesting thing is that most people display incredible strength and resilience. I know this. I know this because I see it every day. The patients and the families that I see, and the stories of courage and strength. And it makes me wonder, why are some people able to cope with unbelievable stressors while other people become completely overwhelmed? Why is that? What drives some of us forward through these stressful times while others may struggle to maintain? Well, I think the answer in part relates to the idea of resilience. The American Psychological Association defines resilience as the process of adapting well in the face of adversity, bouncing back from difficult experiences. Personally, I find the concept of resilience fascinating. I'm really interested in it. Resilience is not just a concept that relates to mental health or medicine. It's something that we know about in engineering and the physical science and physics. Think of a rubber ball. Suppose I had a rubber ball in my hand and I dropped it on the floor. It hits the floor, and imagine this in slow motion, the ball compressing, where the walls of the rubber ball are absorbing the shock from the floor. And then a force within that ball pushes back against the floor, and the ball bounces back up. It bounces up. In a way, it's an example of resilience, where there's these complementary forces of flexibility and strength. Flexibility and strength. And resilience, it's not just something that some people have. Resilience is something within all of us. It's biological. Resilience is something that we think of as a dynamic process. It's not static. It's not that you have it or you don't. It's within all of us. It's made up of, well, it reflects a dynamic and biological process of adaptation. And I'm not the only one who's interested in resilience. Lots of people are. Many people are doing amazing research to understand the science of resilience. Specifically, we're looking at things like the neurobiology, and we've learned a lot. We've learned about brain structure, areas of the brain like the amygdala, the hippocampus, that are associated with the stress response. We know about neural circuitry, neurotransmitters, stress hormones, neural pathways that all have a role in maintaining resilience in the stress response and our ability to respond adaptively to stress. Now, we're not just interested in the neurobiology, but we want to know what helps people to maintain resilience. What helps people to stay healthy in the face of stress? And interestingly, can changing how we think about stress affect how our body responds to stress and our resilience? And interestingly enough, it's true. How we think about stress can influence how our body responds to stress. That the harmful effects of stress that we normally think about are not inevitable. Studies that have looked at individuals who display characteristics of resilience and compare those to individuals who display less resilience, show significant health differences when examined against, in relation to cardiovascular disease and even things like longevity. So, what do we know about resilient people? We're understanding more about the brain, but if we talk to resilient people, or people who demonstrate resilience, what do we learn? Well, there are several, I think, important characteristics or traits that resilient people tend to display. And we know this through studying prisoners of war, through studying people who have survived the 9/11 attacks, who've survived abuse, traumas of all different kinds. And some of these traits include things like optimism, the capacity for positive thinking, a positive attitude. Again, how you think can affect how you feel. Tenacity is another important trait of resilient people. That means determination, not giving up, grit, hanging in there even through tough times. Resilient people also show the capacity to form and benefit from social relationships, social connections. People who are isolated, physically isolated or emotionally isolated, have a much harder time, much more at risk for things like depression or other mental health problems. Resilient people tend to stay connected. Another important dimension includes faith. When we talk to resilient people, many have strong individual values and beliefs regarding faith, whether it's a formal religious belief or a more personal spiritual belief. But faith is an important part of resilience for many people. And flexibility. And I'm not talking gymnastics, I'm talking about cognitive flexibility. The ability to be flexible mentally, to be able to change how you think, which can help change how you feel, and help you to adapt to change and to stress. So, given what we know in medicine and in mental health, I'd like to propose a three-part best practice approach towards maintaining resilience. One relates to lifestyle. Maintaining a healthy lifestyle referable to getting enough sleep, eating a healthy diet, getting exercise, staying connected to friends and family. These things form the foundation for a healthy life, right? And help us to manage the stress of life much more easily. When we're overtired, when we're not eating well, when we're isolated, it's much more difficult to handle stress. Number two, optimizing your medical care. And this is certainly relevant for optimizing and staying active with any care or treatments relating to your melanoma, but also for other things. If you have diabetes or chronic pain or some other health condition, it's important to stay on top of that, right? Because if you have, say, chronic pain and it's not medicated or treated well, that adds suffering to your life. That's another level of stress and strain. So optimizing your medical care means taking the right medications for that pain, making sure that the symptoms are reduced as much as possible in order to reduce the strain and the suffering and the stress that might go along with those problems. And then the third domain relates to how we think, developing healthy habits of the mind that can facilitate healthy coping and resilience. And there are several habits of the mind that I'd like to draw your attention to. The first one relates to this relationship between how we think and how we feel and how we act. Our thoughts, our feelings, and behavior, they're all connected. You know, how we think, our beliefs, the words that we use when we're thinking about things, your answers to me about what autumn means to you, those thoughts help shape how you feel, your emotions, right? Your feelings. How you think and how you feel help shape what you do, the decisions you make, the choices you make. But also what you do, like coming here tonight. You made a choice of coming here tonight. That affects how you think and how you feel. When you go for a walk, on a beautiful fall day, or go apple picking on that beautiful fall day. You may clear your head. You may get some distance, healthy distance from some of the things that are stressful in life. So the relationship between, or being aware of the relationship between your thoughts and your feelings is important. That's one of the unique qualities of being human, isn't it? We can think about our thinking. We can notice how we think, and we can also change how we think. Mindfulness is another habit of the mind that's received a lot of attention lately. We read about it in magazines, we see it on TV, radio. Mindfulness relates to the idea of being in the moment, being present, with an attitude of acceptance and openness. So often, especially when stressed, we're thinking, always thinking about what happened, thinking about what might happen. So many of the individuals that I speak with who are coping with cancer are worried, rightly so, about what might happen in the future, right? How many people have those worries? What's going to happen next? It's only natural. It's only natural. But when we're thinking about those future events, those events that we really can't control, it can add stress to our life. That's where anxiety can come from. Dwelling on the past can lead us into depression. So a strategy for handling or coping with the uncertainties of the future, or in accepting what's past, is coming to the present, being mindful of the present. So again, noticing how you're thinking, coming back to the present, because this is the time that's in your control. This is where happiness and love and joy can occur, now, in this moment. Another habit of the mind relates to how we cope and making conscious decisions about how to cope. I just mentioned before about how future events are not always in our control. And inevitably, in any stressful circumstance, there are aspects of the stressor that are in our control, that we have some influence over, and there are parts of it that we just can't control. For problems that you can control and fix, you're better off fixing it, right? Fix a problem, solve a problem. Utilization of things like problem-focused coping strategies, of gathering information, making decisions, resolving conflicts, setting goals. Those are examples of problem-focused coping strategies to deal with things or to correct problems that are in your control. But what if it's not in your control? Are these strategies going to help? Can you solve the problem if you can't control it? No. In fact, trying to fix something that you can't fix can only lead to more stress. So, the next best thing when you have a problem that's beyond your control and you can't change it, the next best thing you can do is change how you think about the problem. Because remember, changing how you think about a problem can also change how you feel about a problem. So that's the use of emotion-focused coping strategies. So being flexible, again, is important. Knowing and understanding what can you control, what can you not control, and making good decisions about how to best cope with it. Can I fix it, or do I have to find a way to think about it differently and find a way to accept it, or at least to manage my reaction to it? And then the other habit of the mind that I want to emphasize relates to being mindful of meaning and purpose in your life. We are so fortunate to be alive, to be surrounded by friends and family, people who care for us, people that we care for. Cancer can be a very, very challenging thing. We all know that. And sometimes it may challenge your ability to remember what is important. Well, sometimes it may do just the opposite. It may bring right to the forefront those things that are most important. But again, what's in your control? Well, how you live. And asking yourself, what's my purpose? What's meaningful for me? What are my core values? And trying to live a life that's consistent with those values and priorities helps bring meaning and quality to life. And resilience. So, let me sum that up. 3 basics. Eat well, exercise, and rest. That relates to the lifestyle. Be active with your medical care and optimize the medical problems or symptoms that can be optimized. How you feel physically affects how you feel emotionally. So it's important to stay on top of the things that you can influence. Make time with friends and family and accept help from others. That's the part of staying connected to other people. Actively problem solve the things in your control and find a way to accept and learn to adapt to the things that are not in your control. That relates to how you cope. And finally, stay connected to the things that bring you meaning and joy. Because that's where the good stuff is, isn't it? That's where the quality of life is. And it gives us some balance from the stress and helps us to bounce back and to be resilient when we do these things and stay connected to them. So I hope that was helpful. Thank you very much for listening.
So the next speaker is Dr. Ruth Halaban. Dr. Halaban is a distinguished scientist who's probably contributed more to the understanding of biology and melanoma than most people in the world. She's been the leader of our Skin Sport program, which is our major research program in melanoma for the past 10 years. And she's going to talk about what we're looking for in the research in melanoma. Ruth?
Thank you. Shall I put this on?
Give a pocket.
Thank you very much. Just leave it like that, that's good.
You don't want that?
Oh, yeah, I do.
That's you, right?
Okay, yeah. Well, first I feel like I need a lot of resilience to follow up on all those wonderful speakers that we had today. It was really wonderful. So, in a way, I'm going to represent the health spore in skin cancer in a very minimal way, because obviously I'm not going to talk about everything that we are doing. This is a program that I'm the director, and Mario and Marcus Bosenberg are the co-directors. So, in general, the structure of the spore is that there are 4 major research projects, 3 core facilities, administration, tissue resource, and bioinformatics. There is developmental research project, and there is some educational activity, especially outreach with patient advocate. When I started here at Yale, which was about 40 years ago, you can tell from my white hair, there were about 2-3 people working on melanoma. And when we received the spore, which is funded by the NIH, we have now, first of all, involvement of about 14 different departments at Yale. What we had before was dermatology and pathology. That's the only place that anybody studying melanoma. Now you have 14 different departments. And the number of investigators is above 60. So it has been really tremendous. And there is going to be continuation, and Harriet is involved in the continuation of the sport, the next round. So what are here are some of the major questions that we ask. You heard a lot about sunlight and UV light. So sensitivity to sunlight. One of the questions that we ask is, can we tell a person who is susceptible to one that is resistant by changes in the DNA that have been accumulated along the line with time? We are all exposed to sunlight. Some of us develop and some of us don't develop melanoma. Is there anything that I can take the DNA and test it and say, watch out, watch out, watch out, you are a susceptible person? A lot of questions are being asked about immunotherapy and what's happening with the tumor, the microenvironment, and the interaction between the three that responds to therapy. And this is highly complicated subject that I will not elaborate on, but it's very critical for the sport. We are interested in melanoma diversity, and I'll describe that a little bit. What is the molecular diversity of melanoma? Not every melanoma is the same. What is it? Why some respond to drug and some don't? And we are also interested in the mechanism of resistance. What causes a tumor to continue proliferating whereas others are just killed? So regarding the sunlight, I would like to mention work that was done by Doug Brush. And if this is the major lesson that you will take with you home from this talk, it's really very important. So essentially what Doug found is that melanin indeed protects us from sunlight, but it's also carcinogenic. And essentially, the effect of sunlight doesn't stop when you come back from the beach or from the tanning booth. Essentially, there is a process of damage to the DNA that continues after sunlight. And this process, interestingly enough, is produced by melanin. The melanin itself undergoes chemical excitation and continue to send all kinds of harmful messages to the DNA. And this is interesting because actually it doesn't happen in albino individual or in albino mice. It would happen in black, but not in albino mice. So there is a need essentially now for preventive tools such as the evening after sunscreen. And just to indicate that this work that was published about a year ago had a tremendous impact. Doug was approached by essentially all newspapers that are around, all major newspapers. The work was cited by scientific news. The popular press talked about it. And he got even awards that from the Cancer Center and as a keyword award in the American Society of Photobiology. Other impact, L'Oreal, the company that makes sunscreen, is now funding Doug to find out various chemicals that will dissipate this effect of the after dark effect of melanin. And he raised the subject to such a great interest that there is going to be even a special meeting in one of the best spots for science in the United States, which is called Spring Harbor. So, the other subject that I would like to cover a little bit is the molecular diversity of melanoma. So first again, I would like to say that this work was done by dedicated patients, surgeons, oncologists, pathologists, basic research, and scientists. It's a community activity. It involves sophisticated bioinformatics, and it involves a very important resource that we have here at Yale due to the sport, which is the specimen resource core that collects and stores melanoma and blood from patients that come to the clinician or to the surgeon. So altogether, for example, for the past 10 years, we collected 800 melanoma tumors and 600 blood samples and 1,000 normal skin. And each one of these components was important and is important for the next step is to identify the variability in melanoma. Because what we are comparing essentially is, okay, I'll stress that, what we are looking for is to identify changes that happen in the tumor that affects the protein and change the function of the protein. So essentially, just in a nutshell, essentially DNA, that we all know by now what it is composed of protein coding segments that are called exons. And what we are looking for is essentially changes in this region that happen and change the structure and the function of the protein. As you can see here, the human have about 180,000 exons, so that's a lot of work, and it's making about 1% of all the DNA in the human body. So this technique of essentially finding differences in exons in tumors compared to normal DNA was done on 400 patients, which help us identify what we call somatic mutation, changes that are only in the tumor and not in the normal DNA. And changes can happen and have no significance, no effect whatsoever. So we have a special kind of bioinformatic tools that analyze the effect, the change in the protein, to see whether or not it has any structural, any functional, that can affect the process that's going on in the tumor. So what are the results? Essentially, we identify genetic and genomic changes in tumors that are associated with the malignant state. The important thing is that the changes may happen at low or high frequency, dependent what kind of tumor it is. So it was mentioned the tumor exposed to sunlight, for example, has many more changes compared to tumors that are ocular or acral that are not exposed to the sun. Some changes are common. Let's say what I call common is above 1%. And some are more unique to different tumors. And the important thing is that there are also differences. A person that come up with more than one tumor, there are differences between the tumors arising in the same patient. So what are these mutations does? And essentially, Dr. Arian already mentioned it, essentially we have the normal melanocytes in the skin. They can be sparse like in regular skin, or they can be more concentrated as in nevus. And these are the melanoma arise about 65% from here and about 25% from here. As mentioned before, and I'll come back to that, the nevus cells may have a BRAF or NRAS mutation, but they are nevus cells, they are not malignant. So this is another take-home lesson, is that a BRAF or an NRAS mutation that I'm sure you heard a lot about it by now does not change normal melanocytes to malignant. You need more than that. And essentially this is what the other mutations do. They allow the cells to proliferate without control, without environmental input. They start to move from one place to another, they start to move some more, and then the metastasis. So all these activities essentially are allowed, are facilitated by the changes in the DNA that the tumor gets while it's being formed. So here again, the slide that Mario showed. What you have here, each one of these lines is a tumor, and what you have here are tumors with the BRAF mutation. By the way, about 40% of tumors have BRAF mutation. And then here you have BRAF with NRAS mutation, and you can see that very few, there is only one or two tumors here that can have both. Usually they are mutually exclusive. And then there is another group which is the NF1 mutant. This is one of the latest discovery that essentially melanoma can be defined, like 85% of melanoma, you can see that, can be defined by either BRAF mutation, NRAS mutation, or NF1 mutation. And then we have a fourth one that is, we call it now triple wild type. And over here you have the other mutations that are more or less frequent in melanoma that may occur in some and not in the others. So, another interesting observation. As you all know, we have the redhead, blue eye, fair skin phenotype that is susceptible for melanoma. Usually this phenotype is due to a receptor, the melanocortin receptor. You might have heard about that too. This is in short called MC1R. So the melanocortin receptor have variability, and now this is genetic, that affect the color of the eyes and the skin and the hair. So we call this variant red hair MC1R variant. Now, as it turned out, tumors derived from patients with the red-haired phenotype have about 42% more mutation compared to those that have, so to speak, the normal allele. And this mutation burden is comparable to that associated with additional 21 years of aging. So that's maybe another thing you can remember is that having the redhead fair skin phenotype has a very significant effect on the number of changes and mutation that a person can be developed. Okay, so now I'm coming to what was mentioned also before with precision medicine. So we know that. I have a computer that is full of Excel files with all those mutations and changes listed there, and I can look at it forever. Can that affect the patients? And this is one of the things, this is the next step that has to happen, that genes and environment and lifestyle have to come now to take it to the clinic to take care of patients. This is the medical decision and practice that has to be now be installed. So essentially, I'll go a little bit faster. What we want is essentially to incorporate melanoma genetic into patient care. What needs to be done is essentially to sequence. So what we have done, we took the DNA and we subjected it to what is called next generation sequencing. That, I believe, has to be done now for every tumor that we get out of the clinic. And one of the main reasons, obviously, is to know what's wrong. But another very practical reason, and I heard it today from Ira Melman from Genentech. He is in charge of essentially the research in Genentech. That's what they are doing now. They are looking for variants. You see, the protein that is being made is different. You didn't have it before. And it can be used as a vaccine, except that, as I indicated to you, there cannot be, I wish there could be a vaccine against the BRAF mutant or the NRAS mutant. Then we would all be quite on a good future. However, the immune response is very unique, and each one of you might have a different antigen that raises the immune response. So that means in order to find it, we have to sequence every tumor, and then obviously that has to be analyzed to find out what exactly the protein that is antigenic there. So as I indicated, we have 85% of melanoma essentially can be recognized by this mutation. This mutation by themselves will not make melanoma. You need the helpers. And how can we get that to the clinic? One way is and again, some people are working on that, essentially to develop what is called liquid biopsy. To take DNA from the tumor, you have to have an operation. Operation is a major event. An alternative way of doing it is to find the tumor DNA in the blood. And it turns out that it can be done. You think about the blood, how much tumor DNA can be in the blood? Very little. Less than 1% than total DNA can be in the blood from the tumor. But it's there. And now we have a very sophisticated way of analyzing it. So essentially, the idea now is to bring genetic to the clinic by collecting blood at different time point, capturing tumor DNA. Use this mutation that we know exists. Use this to capture the DNA, the tumor DNA from the blood. And then identify the mutation, obviously. Now, this can give you different information. One is tumor load, and it was shown already, by the way. By using the BRAF mutation or NRAS mutation, it was shown that you can estimate the amount of tumor in a person even before various imaging by just doing that. Because more tumor DNA in the blood, higher load. Another thing is the molecular signature, the tumor evolution. Talking about evolution before. Novel mutation, emergence of new tumor clones according to the new mutation, and drug resistant. And as I mentioned before, neoantigen that's very important and can be useful. But all this information, it's not easy. It's actually, we need computer to analyze it. It cannot be done, in 10 minutes just by looking at that. And it turned out that in New York, there is a computer called IBM Watson Cancer Genome that essentially is dedicated just for that. The computer takes the genetic profile, the information about the medical literature, and the personalized treatment option, and analyze it. And essentially it was done. It was done by Robert Arnell, who is established the New York Genome Genome Center, and he applied it to 30 patients with glioblastoma, and essentially developed personalized treatment to each one of them, and that's a publication that will come out soon. So this can be done. And to this center, essentially, there are now 14 leading cancer institutes that collaborate, and I think we should get there too. Okay, now I'll finish now with the cancer moonshot that you might have heard before, and I'll not go through each one of them. You can Google and find out, but essentially the cancer moonshot that Biden started, they have now listed at least 10 different priorities that should be obviously implicated in the coming years. And some of them is essentially what I just described to you. So this is the Yale Sport team. These are our surgeons and clinicians, and several of them you heard today. This is our bioinformatics, basic research that we do. The funding is from the NIH, from Melanoma Research Alliance, Gilead Company, Rose and Jeremy Meyer gift, that's our patient advocate, dermatology, and Yale. And many thanks to all the patients. Thank you.
Yes. There's one more speaker. The last but definitely not least, Dr. Harriet Kluger, who's a professor of medicine and has a longstanding interest in developing biomarkers. This is the blood tests and the tissue biopsies that we do to try and figure out how to match the best patient with the best treatment, and also to determine, to some degree, the outcome of the disease, how well people will do, and whether you need treatment or not. So with that, Harriet, I'll please up and enlighten us on this very important topic.
Thank you. And I realize that I'm the last person between here and going home. So hopefully I won't put everybody to sleep. And actually I have to start off with a bit of a confession. I was going to start by saying we don't really know how to personalize treatment for melanoma. Our biomarkers still are in their infancy and we've got a long way to go. But after listening to Dwain's talk, which was so uplifting, I decided we're going to give a positive spin to the end of the evening, and I will overemphasize the progress, but again, I might be off topic relative to what's written in the slide. So without much further ado, I guess the question is, well, why do we even need biomarkers, particularly for patients who've got advanced melanoma? So firstly, well, what is advanced melanoma? As you've heard before, melanoma starts in a certain place. It can be the skin, the eye, or the mucosal area, and a couple of other areas. Once it's resected, sometimes we find it pop up in other places, and at that point we have to give what we call systemic therapy, which means therapy that goes in either by mouth through pills or by IV, so that it can get to all the different parts of the body and catch those little dandelion seeds that Dr. Arian was showing you earlier if you were paying attention. So we can either treat the dandelions themselves, the cancer cells themselves, or we can treat the soil around them, poison the soil so that they can't grow very well. And those are the two major approaches that we've taken in recent years. So actually, I'll try to use the point here. What actually is the problem? Well, maybe it's not really a problem, it's an issue. That's to reflect Dwain's positive attitude over here. So until 2011, we didn't have a problem because there was really it wasn't very much that we could do. There were only 2 FDA-approved drugs, and they were high-dose interleukin-2 and dacarbazine. The response rates were low. Since 2011, this whole list of drugs has been approved over here, and then there are a whole bunch of combinations. So how do we actually decide which patient to give which drug to, and whether we give 1 drug, 2 drugs? We know the more therapy that we give, we might have more activity, but at the same time we'll have more toxicity. Maybe some toxicity is actually worthwhile because there are a few familiar faces over here in the audience who've had toxicity of some or other sort, but have overcome the toxicity and actually doing very well and going about and living their lives now. So it's very hard for us to select what drug to give to what patient, and we have a long way to go to learn how to actually do this. But this is essentially what we're really grappling with. As I said, we could either target the cancer cell or we could target the immune cell. So there's not the biomarker type of selection, there's the interpersonal type of selection. Well, does the patient have a specific mutation? Normally we will go with targeted therapy at some point if a patient does have certain mutations, particularly ones like BRAF that Ruth mentioned. The advantage to targeting the cancer cells is that these drugs often work in a high percentage of patients, provided that they have the mutation that we need. They work quickly. The disadvantage is that the cells become resistant with time to these therapies. Another advantage is that they pill form. They're relatively easy to do. For immune therapy, we don't require specific mutation, at least as far as we know at this juncture. Sometimes they take time to work. But the nice thing about it is when they do work, they can work for a long, long time. And pretty much all of the folks around here who we've been seeing for many, many years have been treated with immune therapies. But they may work in a small percentage of patients. Some may work in more, some in less. And the question is, which one do we choose and for whom? So I'm going to start off just with a couple of minutes talking about targeted therapy. So this is a little cartoon here of a cancer cell. This is the cell membrane around it. You have these little receptors over here, and they're things that make these cells grow and divide. And there's all sorts of signaling molecules that talk to each other within the cell. Now, if they become mutated, they might be turned on forever. And the one that Ruth mentioned that as being most common is BRAF. So it's mutated in around 40% of melanomas. And when it's mutated, it will tend to signal and make the cell grow and divide continuously. The second most common one is NRAS, and then there are the other ones that are rarer. So not only do we need to know what the mutation is or what protein the mutation is in, it's also important to know the specifics of the mutation. So I'm going to show you an example over here of why this is important. So in actual fact, BRAF mutant melanoma is the only instance in which personalized medicine actually works in melanoma, because the response rate in patients who actually have the mutation is around 90%. It's a very easy test to do once we have the tumor. It's clean. There's no false positives. Either the patient has the mutation or doesn't have the mutation. And then there are a bunch of different drugs and drug regimens that we can use in a patient who actually has the mutation. So we did think that you had to have this mutation called V600E. With sparing you the details of what that means, the initial trials were specifically for patients who had this V600E mutation. And there was one patient who went down, I think it was Dr. Sznol's patient, went down to New York to be enrolled in a clinical trial for patients with these BRAF inhibitors, and Ruth had the tumor in her lab. And Ruth said, nope, this guy has a different mutation, V600K. And until Ruth did this, I don't think we really knew that V600K mutant melanomas can also respond really well to this class of drugs. So sometimes we learn things by serendipity, and the other thing I want to bring up here is a little note of thank you to everyone who actually provides tumor samples for Ruth's lab, because this is how we find these things. So that is really, really important, and I would encourage everybody to continue to donate blood and tumor. Even if it doesn't help you, it might help the next person, and I think this little finding might have helped a lot of people along the line. So what about other mutations? There's the NRAS mutant melanomas. Those we have a more difficult time to target because we don't have a specific drug that can inhibit NRAS protein. But we're working on that. And then there's another one called c-KIT, which is incredibly rare. Less than 1% of the melanomas have mutations in c-KIT. But there is a drug that targets c-KIT, and it works pretty well when the mutation's there. What about immune therapies? So there are no known really good biomarkers for immune therapies, and that's one of the problems that we grapple with every day in the clinic. We use a bunch of different types of immune therapies. The older ones, interleukin-2 and interferon. People spent a few years trying to identify biomarkers predictive of response for these therapies. We're no longer really spending much energy or resources on it, so I'll move on. Adoptive cell therapy is a type of therapy that was done at the National Cancer Institute for many years. We now have our second clinical trial over here doing a certain type of adoptive cell therapy, and we actually have treated our third patient on it. We sometimes can, in certain types of adoptive cell therapy, we can predict response. The vaccine therapies, there's no predictor of response to that. And I'm going to just spend the next few minutes talking about the immune checkpoint inhibitors, because these have now become the mainstay of therapy for metastatic melanoma. And I'm going to show you a little cartoon in a minute just to describe what we're talking about. So I took this cartoon from Dr. Sznol a few years ago. And just to give you some idea of how things have changed, pretend this is a T cell and this is a cancer cell, and they kind of talk to each other when they come into contact. And a bunch of little signals are turned on and off, and we call those immune checkpoints. So either they can stimulate or inhibit the T cell. So this one, CTLA-4, is the target of the first drug that was approved, called ipilimumab or Yervoy. And the other one, PD-1, is the target of nivolumab and pembrolizumab, also known as Opdivo and Keytruda. Sorry, the names are really confusing. But these were the two checkpoints that were studied the most in the beginning. But there are a bunch of others that we already knew about at the time. And now fast forward to 2013. This is a little cartoon that I took out of a paper that was written by Liping Chen, one of our scientists. And here he described all of the checkpoints that were known at that time. And as you can see, there are a whole lot more of them. And I put little circles around some of those that are targets of drugs that are actually currently in development. So this class of drugs is going to be the backbone of everything that we do in cancer therapy, not only for melanoma, but also for other diseases. And I think primarily PD-1 is going to be the drug that we use as the backbone to which we add other drugs afterwards. So the question is, well, can we predict response? So actually, ignore this line, is from an old slide where I say low response rates, because it used to be like that with the first drug when we used it by itself. So can we predict response to ipilimumab? Well, there were a number of attempts, and I won't go into the details, but there was a study that was published in 2011, and the Yale group contributed samples for the study, where we showed that we could somewhat predict response to ipilimumab, but we don't really use it anymore. and we're not trying to develop better tests because we're no longer really using ipilimumab by itself very often. We do it rarely. So now we need to move on to the next drug that's been developed, and this is the inhibitors of PD-1. So PD-1 targets, the PD-1 inhibitors, target a little molecule that sits on the T cells called PD-1, and that interacts with a little molecule that sits on the tumor cells called PD-L1. So if we can measure PD-L1 in the tumor, the question is it there? If it's there, will the patient be more likely to respond? So the answer is yes. When you give nivolumab, if you have the ligand, you're more likely to respond than if you don't. But look very carefully at these numbers. There were 3 out of 21 patients in this particular trial who did not have the expression of this PD-L1 marker, who did respond, and of the 17 who had it, there were 7 who did not. So it doesn't necessarily follow that everyone who has the biomarker is going to respond and everyone who doesn't have the biomarker is not going to respond. When we give the two drugs together, ipilimumab and nivolumab, you see that the biomarker is actually becoming less and less important. This is a study done in my lab by one of my postdocs, where she collected specimens from patients who got ipilimumab and nivolumab in combination. And the question is, well, when you have PD-ligand 1 on the tumor, are you going to be more likely to respond to the combination? But in actual fact, it was very interesting. We found most of the PD-ligand 1 was expressed in the immune cells, not in the tumor cells. So we were actually looking for it in the wrong place all along. And it turns out that when it's present in the immune cells that are infiltrating the tumor, you're more likely to respond. But just like the previous study, even if you do have it, there was 12% who didn't respond. And among those who didn't have it, there was 45% who did respond. So it's not a clean enough biomarker to use for selection of who you're going to give two drugs to versus whom you're not going to give it to. There are a lot of efforts ongoing by other groups across the country. I think this is one of the more active areas of research. The UCLA group showed that it's actually a lot more complicated than just having this one marker within the tumor cell or within the immune cells. And they found that patients who did not respond had less immune cells, as one would expect, because there are less things to stimulate over there. But also, on treatment, the patients that did respond, you had massive expansion of the amount of T cells that were sitting within those tumors. So that's a really good indicator, but we can't keep biopsying people. So as Ruth said, we need to have biomarkers that come out of the blood because they're just much more practical. But once again, over here, you have responders who do have the biomarker and non-responders who don't. And, sorry, the other way around. And it's just not clean enough to use for patient selection. There was another study that was just published in Cell this year where they looked at the tumor burden, and this reflects on what Ruth was talking about before. If you take a tumor from a patient, you can just measure the sheer number of mutations that they have. And the greater the tumor burden load, the more likely a patient is to do well on inhibitors of PD-1. But still, once again, you have patients who have high tumor load who still don't respond, and low tumor burden who do respond. So it's still not good enough, and we still have some work to do to get there. But as Dwain would say, this is an opportunity. So I'm just going to summarize. We won't go through the slides all over again, but we have all of these tests that we're doing to try to identify predictors of response. We're not there yet. We can't really use them in the clinical setting. Really, the only thing we can do is to retroactively analyze specimens from patients, and I thank everybody who's been willing to donate their tumors for it. And also to point out that one of the other challenges that we have is that the drug development field is moving so fast that every time we think we've found a biomarker for something, it's no longer relevant, just like the biomarkers that we thought we found for predicting response to ipilimumab, maybe we don't need them anymore. But that's a very good problem to have. And I think that the other problem we're going to have going forward once we have a bigger menu of drugs that we can use is determining the co-expression of the targets of each one of those drugs. So the field is becoming more and more complicated, and we're going to need more complex computer programming to determine who we should treat on what regimen and whom not. So I'm just going to conclude in a few sentences that therapies for metastatic melanoma have dramatically improved in recent years. As Dr. Sznol showed you earlier, those 5-year survival predictions are much better than they used to be, from a 5 or 10% many years ago to what we think is now going to be 50% and hopefully more. But more than ever, we need biomarkers that are predictive of response because we've got more choices. The best tests that we've had to date have been the mutations, and specifically for BRAF. But we do need more sample collection. We need to do much more work on this, and careful analysis of the data, sharing of reagents, sharing of samples between different sites. That'll also help, and the NCI is encouraging us to work together to do that. So I'm just going to end up with an acknowledgement. We have the entire Yale clinical research team, our pharmaceutical collaborators, research collaborators, and of course all of the patients and families who are willing to help us do this work. And then there's the larger melanoma research community that's often